And with this, we come to our last talk, Wilson Araujo, on improving therapeutic responses for high-grade glioma using single-cell genomics. Luis,
good afternoon. First of all, I would like to thank the scientific committee of the HCA Latin America virtual workshop. Thank you very much. My name is Wilson. I'm a professor at the University of São Paulo, and I'm here representing the Guarapuava Institute for Cancer Research and the Genomic Network of the state of Paraná. I'll start my talk by presenting the city of Guarapuava, which has a population around 180,000 people and is located in the center of the Paraná state. The city has an excellent quality of life and is going through a moment of great development due to a local public-private partnership. The goal of Guarapuava is to become, in the next 10 years, a reference center for cancer research and of development of technologies applied to digital health. Today, I'll present two pilot projects. One that aims to use a single-cell approach to investigate in vitro subpopulation of tumor cells resistant to chemo and radiotherapy. And another one is to analyze the genetic determinant of immune phenotype variants in a cohort of healthy individuals. The experimental design for the project one begins with the selection of 10 GBM samples, could be another tumor. With a fraction of tumor will be performed RNA-seq, whole exome seq, copy number variation, and attack-seq, and perhaps the spatial analysis of single cells. Next, we'll do the dissociation with another fraction of tumor to perform phenotyping, single-cell RNA-seq, single-cell copy number by RNA-seq, and a single-cell attack-seq. And another fraction here in B3 will be used to generate the GBM organoids, which will be treated with drugs and analyzed again for the same single-cell genome protocols before and after treatments. To the preliminary results, we are using public data from single-cell RNA-seq and to training the bioinformatic analysis. The idea was to identify cell population with active EMT mechanism. For that, we use information from a study published by our group, which demonstrated that HOTE, a long non-coded RNA, was required to trigger the mechanism of EMT and metastasis. The UMAP analysis in A shows a cluster in orange of the tumor cells and another cluster of the non-tumor cells dyed in black, pink, green, and red. In B, we have the subpopulation of both cell types, tumor and non-tumor cells. And in C, we can observe HOTE's expression in a particular subpopulation. Interestingly, in D, we have the absence of E-cadherin expression in the same subpopulation. We know that the loss of E-cadherin expression could be the first step in activating the EMT mechanism, of course, along with the expression of other genes like snail, twist, etc. Finally, in G, we applied an EMT gene expression signature, which was enriched in the same cell subpopulation with expression of HOTE. With this result, we can strongly suggest the HOTE's correlation with EMT and metastasis. EMT is one of the mechanisms that could be analyzed to find the subpopulation of GBM associated with resistance to chemo and radiotherapy. The second pilot project will be part of the genomic study of Guarapuava population. The project will be launched in October next month with the aims of sequence the complete genome of 1,000 healthy individuals from a select sample of 5,000 people. We want to take advantage of this project and analyze the genetic determinant of immunophenotypic variants in a group of 30 healthy individuals. Our intention with these two proposals is to participate in HCA Latin America as a training and research center in single-cell genomics. So, what can we offer as a member of HCA Latin America? We can offer the institute infrastructure and the expertise of 150 research distributed in 13 institutions, which are members of the Paraná Genomic Network. Both the institute and the Genomic Network are coordinated by Professor David Livingstone, who is designed to be joined HCA Latin America. We also have the institutional commitment of the Mayor of Guarapuava, César Silvestre Filho, and the General Superintendent of Science and Technology, Professor Aldo Bona, to financially support the two pilot projects in co-partnership with the HCA. The Guarapuava Institute for Cancer Research was opened in August last month and consists of a scientific platform for genomic research. The institute equipment base range from DNA and RNA extractor here to Promon from 10x Genomics and NovaSeq from Illumina. As I mentioned before, the Paraná Genomic Network has 13 research institutions spread throughout the state. Under the coordination of the Cancer Institute, the Genomic Network can develop a project that covers several areas of interest of human health, such as sports genomics, oncology, Mendelian disease, metagenomics, nutrigenomics, pharmacogenomics, genomics epidemiology, and forensic genomics. Thank you very much for your attention.
Thanks a lot, Wilson. Great talk. Looks like a great institute and a great infrastructure to do research. A lot of single cell equipment plus sequencing power. That's a very good combination.
Yes, that's the idea.
I had some question about the single cell, like the wealth of single cell data sets you're generating from RNA to ATAC to copy number variants. So what do you think would be the driving data type in your analysis to understand glioblastoma or glioma? And
yes,
how do you plan to combine those together?
Yes, the idea to use especially the single cell RNA-seq to combine the gene expression, use the same data to try to analyze the component variation. It's the algorithm published by Tirosh, and my PhD student used to analyze is work very well. I think it's possible to.
Okay. Yeah, there's a question in the chat. So people are impressed by your center and by the infrastructure you're providing. The question is, how can people best approach you, and if you're open for collaboration?
Yes, the institute is creating a scientific platform for genomics analysis. It includes single cell analysis, and the idea is to work like facility to provide analysis for the people from the research from the Paraná, and of course for the rest of the Brazil if the US. But now the next step is to start to training people in genomic analysis. We create the infrastructure. I think it's more easy, and we only have to have the money. I think we now we have, and the next step is a little bit difficult, is taking more time, is to training people in bioinformatics analysis.
Yeah, training in bioinfo is key. I agree. Do you still see me and hear me? Because I lost my video. You still see me?
Yeah.
Okay, first, then just keep talking. Thanks a lot, Wilson. Thanks to all the speakers in the session. It was great listening and seeing the advances that are done or like presented in the session. And with this, I would thank the speakers, the audience, and I would hand over to Lucio for some closing remarks. I think we kept the time. It's now almost 11 o'clock here in Spain. Lucio, I would hand over to you, and yeah, thanks a lot and see you soon.